Hello students, we had talked about interphase in the previous lecture. Now we are going to deal with the M phase basically. That is the main study material under this topic basically. Interphase though is, but M phase happens to be the most important phase in cell cycle. So the question that is that why is M phase important? We had discussed that the cell has to divide itself into two daughter nuclei. Now you might be getting bored of this thing, but this is the only process which we are studying. How it is happening, that we will study. The two daughter cell division which divides these two daughter cells, which divides this cell into two daughter cells further, is manifested during M phase. So certainly it is very important. Ultimately, after cell cycle, you need to see two daughter cells. Which process is giving rise to those two daughter cells? The interphase does take place. It is preparation for M phase only. But what process is responsible for giving two daughter cells to you in the result is basically the M phase. That is why it is important. Moreover, secondly, what is more important in M phase is that we can microscopically see the behavior of cell in cell cycle, the simple microscope, I am not talking about electron microscope, under M phase only. The microscopic study of cell cycle could be done in M phase, because at that stage, please pay attention, at that stage the genetic material is completely condensed and visible. I am going to repeat this sentence time and again. So this is important, that the genetic material gets condensed and is visible. It means that you can somehow see DNA. Seeing DNA is not possible under microscope, even the simple one. It could be seen under different specialized microscopic techniques. But when we see it under simple microscope, we do see the DNA in condensed form in chromosomes. So we need to know what is a chromosome. You might have heard this term chromosome many a times, but let's see in terms of DNA what is a chromosome. If I see a chromosome, it is basically condensed DNA which is arranged like this. Alright, you would study how the DNA is coiled and arranged on a chromosome. Never before you would have understood chromosome in this manner, that chromosome basically contains the DNA. Now you studied S phase. Supposedly this was the actual chromosomal content of a cell. This was the actual blueprint on which the cell worked, the parent cell I am talking about. So after S phase, what would happen? A similar chromosomal material would be formed and it would be in close proximity to this only, attached to a point known as centromere. This structure of chromosome and its parts you have done in your previous classes, but over here see it in context of DNA. Now this was the original DNA. Over here in S phase, the entire DNA content is to be duplicated. That means a similar content is to be formed during the S phase. So what we have as a result of S phase is that this much, double the genetic material has been formed. Now see the importance of interphase. Supposedly, take it in terms of grams I would take. Supposedly you had 4 grams of DNA when the parent cell started its cell cycle. When the parent cell started its cell cycle, it was 4 grams. After the completion of S phase, the genetic content would have gone 8 grams. Now because we are dealing with chromosomes over here, so we need to pay attention to the chromosome number as well. The genetic content has doubled, but a chromosome is identified on the basis of its centromere. So even if there are 2 sister chromatids which are attached on a chromosome's centromere, it happens to be a single chromosome. So at the end of S phase, even after getting double the DNA content, the number of chromosomes remains same. Now this DNA content has to be separated into the both daughter cells which would be formed, and that is what M phase is going to do. First of all, the genetic material in S phase is not at all condensed. So what is the term basically for that? That is chromatin material inside the cell is scattered. It's just like you are having threads and they are thrown everywhere. After some time you arrange them into specific bundles and they become chromosomes. You can, draw a parallel between it. Chromatin material is the simple material which is scattered inside the cell, which is not condensed, and if it is scattered, it would not be clearly visible. This chromosome thing is also visible. If you see under microscope, it is visible like a dot. When you have to increase the magnification, then you see this sort of structure. So you can very well imagine, if the material is not condensed, it won't be visible under the microscope. So the chromatin material starts condensing, it forms the chromosomes, and now what is the purpose of M phase? Is the separation of these two sister chromatids in such a way that one of it goes to one daughter cell and the other one goes to other daughter cell. Am I clear with it? I would repeat it, that basically the chromatin material has to be separated into the two daughter cells. The chromatin material which has been doubled during the S phase. Now it would be easier to divide the chromatin material in both the cells if both of them, if the chromatin material, not both of them, if the chromatin material is arranged in a specific way. Now that arrangement is possible when the chromatin material gets condensed and we see chromosomes. So make one thing clear in your mind, that chromosome-like structure is not present in the cell for 95% of its lifespan. It is only during the M phase that we have chromosomes present inside the cell, and we time and again use the term there are this much chromosomes inside human cell. It is only during the M phase that we talk about chromosomes. So now this M phase would be dedicated to the separation of genetic material into the two daughter cells. Now you realize why M phase is important? Of course it is the most important one, because the division process, when I talk about cell cycle, there is a cell division basically, and it is totally because of M phase processes, that is various stages of M phase which are going to take place. Only because of that, the genetic material which got doubled during the S phase would be equally distributed between the two daughter cells. Now we come to the next question, that is, what are the two types of cell divisions? Cell division means M phase. There are two types of cell divisions. First being mitosis. You see a plant, it is a seed in the beginning and it grows into a huge mango tree supposedly. How is that possible? The fundamental unit of life being cell, the cells must have divided. So the division which is responsible for growth in organisms is the mitosis, and the second type is meiosis. Now to understand these in a very clear-cut way, and a clear-cut distinction that you would want from it, that how is this division different from this division? So please keep in mind, this is equational division and meiosis is reductional division. Now as these are simple English terms, so you can associate a meaning with it, that equational, the daughter cells are going to have something equal in them, whatever the daughter cells would be formed, and in meiosis, whatever that thing which would be equal in mitosis would have been reduced. So what thing we talk about when we talk about cell division? It is the genetic material only, because that is the most important thing which would govern the cell. So in equational division, you are going to get the daughter cells which would have equal DNA content. That means the daughter cells would have similar DNA content as it was found in the parent cell. And in case of meiosis, the daughter cells would have half the genetic content as was present in the parent nuclei. Now see again, if I am talking about mitosis and consider it that this is having 4 grams of DNA. Again I am using this 4 grams thing which got changed to 8 grams after S phase. When we are going to have daughter cells, both the cells would have 4 grams of DNA, because in the beginning the parent cell had 4 grams. Don't confuse yourself with the chromosome number in this, because this is the genetic content. That means the chromatin material which got replicated. Supposedly there were 4 strands, they got replicated into 8 strands, and the offsprings are going to get similar genetic content as was present in the parent cell. Now in case of meiosis, similar thing would happen. Suppose this cell is having 4 grams, again I am saying that 4 chromatids are present. After S phase, similar thing would happen, but during mitosis, during M phase, this particular cell would reduce its genetic content into half. is it clear that 4 grams in the beginning, there were 4 grams of DNA inside the cell. Now there is equivalent division, but it, as you can see, this is 2 grams and this is 2 grams. But it has got double in this, it has got 8 grams. So by any means, mathematically also this is not correct. So there are going to be 4 cells which would be formed, and then it would be correct mathematically as well. So first thing that you need to remember is that in meiosis you are going to get 4 cells instead of 2 daughter cells. There would be 4 daughter cells which would be formed. Secondly, in matter of chromosomes. Now, what is confusing in cell division is basically the number of chromosomes. Talk about mitosis first. We did discuss right now that how a chromosome number is restored, how the chromosome number doesn't get increased even after S phase has completed itself. This is mitosis, where I have considered 6 chromosomes, alright. 6 would be there in totality. Then after S phase, what would happen? The chromosome number would remain same. Supposedly this is the chromosome in the beginning. It is, you have 6 such chromosomes in the beginning. After the S phase has been completed, the chromosome number would be same, but they would have an additional sister chromatid. Sister chromatid is nothing but the condensed chromatin material which resembles, you can consider it as a butterfly probably. So the number has not changed yet again. It stays 6 only. It is totally different from that of genetic material because it got doubled. As you can see over here, if this is 1 chromosome that is having this much genetic material, over here you are going to have 2. So the genetic material has doubled, but the chromosome number has remained same. And after mitosis, what would happen? The daughter cells are going to have similar number of chromosomes as the parent cell was having. But this won't be the case in meiosis. Over here, consider it again. First of all, by default, you have to remember one thing, that in any case, there won't be formation of 2 daughter cells. There would be 4 daughter cells which would be formed. Alright, right now we saw that how the genetic material is also reduced to half. Let's see what is about the chromosome number. Here also consider that you are having 6 chromosomes which were like this in the beginning. After S phase got completed, the chromosome number still remains same. There are 6 only. But now what would happen, that the chromosome number over here would be 3 in each case, and hence the genetic material. So over here, why it is reductional? Because not only the genetic material is getting reduced to half, but the chromosome number is also getting reduced to half, because the parent cell had 6 chromosomes in this. This is just for an example. It had 6 chromosomes in it, and the offsprings are having 3 chromosomes, which is exactly the half of what is present in the parent cell. Now, this is the basic difference between these 2 types of division. When we see each division in detail, we are going to understand how it happens, why it happens. How it happens can be taken later on. Why does it happen? Why do we need to reduce the chromosome number to half? The reason is simple, that this division is going to take place when there is sexual reproduction. In sexual reproduction, you know, 2 parents are involved. One would be male and other would be female. Alright? When I talk about sexual reproduction, I cannot miss the mention of gametes. And when we talk about gametes, then meiosis has taken place. So in sexual reproduction, supposedly this is the father. No, this is the mother. This is the father. Father is having 46 chromosomes. Let's take the example of human beings only. We have 46 chromosomes in each cell of our body. Hence the gametes would be formed from the 46 chromosome number, but the chromosome number would be reduced to half. Why? Because if this and this, these are gametes in case of sexual reproduction of humans. If these both happen to be gametes, what would be the chromosome number in the offspring? You can very well make it out that these chromosomes and these chromosomes both would come together and it would become 92. And no human has 92 chromosomes in it. So it would be not a human being. These parents will not have a human offspring. It would be something else. It could be a giant or a polyploidic human, which has never occurred in nature though, because we are highly advanced organisms and we have the ability to do meiosis. Now consider this, that again 46 cells in both cases. If I reduce this 46 to 23 by some method, and this is also reduced by 23 by some method in a certain cell, and both these cells unite by the process of fertilization, the offspring is going to be similar as in the case of parents. Are you getting my point? I'm telling over here that 46 chromosomes were present in father's cell, 46 were present in mother's cells. If they reduce it to half, then the offspring is going to have exactly the same number as both the parents are going to have. So somehow this division is vital for reproduction sexually, that is done by sexual methods involving both the parents, and it restores the chromosome number. It does not lead to aberration in the chromosome number. For that purpose, meiosis is important. And for growth, certainly mitosis is important. Once the offspring has been formed with 46 chromosomes, it has to grow further in size. That would be done by mitosis, and when it reaches its reproductive age, then that offspring has to further produce gametes. That would be done by meiosis. Now let us see what are the stages of M phase. You get to know answer of this, that how is mitosis different from meiosis? How is it different in stages? That we shall see when we see each division in detail. But for once you get to know that one would be reductional and other would be equational division. Now what are the stages of mitosis? First one is prophase. There is nothing difficult to learn these, phase and pro. First one, pro word I told you earlier also that it somehow means which comes first. Then we have metaphase. Meta term, you know where it is used? It is used for intermediate, anything which is intermediate. Then we have anaphase, and fourth one is telophase. Now during these four phases, there is a lot of activity at chromosomal level. Alright. Now what is there in prophase which is different from metaphase? For that we need to see how a cell looks when it has entered M phase under a microscope. What a cell is? It is having a cell wall or the cellular envelope, and the most important part, that is nucleus. So this is the nucleus, this is the cytoplasm. What has to happen? First this nucleus has to divide into two parts, and then this cytoplasm has to divide. So we have two stages in M phase. That means that two major processes which are taking place. One is karyokinesis. Kinesis means division, and karyo word is used for nucleus. Other is cytokinesis. As I told you, kinesis is equal division, or cyto is cytoplasm I am talking about. So first of all, what takes place is division of nucleus into two nuclei, daughter nuclei, and then the division of cytoplasm. This karyokinesis involves these two stages. Alright, these four stages, not two. This karyokinesis process, where the nucleus would be giving rise to two daughter nuclei for the two daughter cells, is known as karyokinesis, which is completed in these four stages, which is followed by the formation of two different cells. When two daughter nuclei would be formed like this, supposedly two daughter nuclei are formed after karyokinesis, then there would be division in cytoplasm as well, and we would have two different cells which would be formed, and they would be daughter cells. Alright, they would resemble their parent cell in any means, because it is exact duplication of cells. In cell reproduction, we are not talking about any sort of variability that can come in cell, but yes, during meiosis, variations can occur at chromosomal level. That means some way or other the genetic material can get exchanged. For now, pay attention to the four stages of karyokinesis. Prophase is the stage when the cell is basically entering the M phase, where the nucleus chromatin material is getting condensed. Alright, this is the cell, this is the nucleus. Over here, the chromatin material is getting condensed. I would make the nucleus larger so that you get an idea that this is the primary organelle which is involved in cell division. Here is the chromatin material. It starts getting condensed during the early prophase stage. Also, you know that this nucleus is surrounded by a nuclear membrane which is in continuation with endoplasmic reticulum, Golgi bodies, which are endomembranous structures. They start getting dissolved. That means the cell would become uniform in terms of chromatin material distribution. This ER would also dissolve in the prophase, Golgi would also dissolve. All these dissolutions are taking place because of the proteinaceous changes and the enzymatic activity which is taking place. So this chromatin material has started condensing itself. There is lot of chromatin material. You can very well imagine there is 2.2 meter long DNA present inside our cell. That cell which we can't see even with our naked eye. So this chromatin material, it starts getting condensing. When the prophase ends, this chromatin material has condensed to a level so that it starts looking somewhat like a chromosome. You can take it like this. This is the chromatin material. The earlier chromosome that I drew was quite good looking than this. It somehow resembled a butterfly. This is scattered everywhere because the nuclear membrane has dissolved. So when the prophase is completed, what happens is that these chromatin material are going to get more condensed. That means it is going to take a better shape as compared to the earlier one. This is going to get more condensed. Also, one remarkable thing that happens, if we are considering plant cells supposedly, one remarkable thing that happens, plant cells have centrioles, you know that. The centriole duplicates itself during the G2 phase. It duplicates itself during the S phase, not the G2 phase. The centriole has duplicated. One centriole comes over here, the other one goes to the other poles of the cell. These are centrioles, okay? I would write C so that you can easily remember it. The centrioles had duplicated during the S phase. They have moved towards the poles. Now, centrioles are microtubules.